Welcome to the Explainer. Right now, as you're watching this, there is a literal high-stakes, microscopic true crime thriller taking place inside your body. It's this continuous life-or-death surveillance operation, and it's happening millions of times a second. Today, we're going to unpack the brilliant, beautiful, and honestly, absolutely ruthless biological machinery that keeps you alive by constantly asking one simple question: friend or foe? Okay, let's dive into this. The absolute stars of our adaptive immune system are the T cells. They act as the ultimate coordinators and assassins for your body. But these assassins have a massive, potentially fatal flaw. They're essentially flying blind. A T cell simply cannot look inside your cells to see if a virus or some bacterium is hiding out in the cytoplasm. I mean, the threat could be replicating right under their noses, and they wouldn't even know. And to make matters even more complicated, T cells can't just spot free-floating threats on their own either. They desperately need a highly regulated system to differentiate between self, which is, your own healthy host tissue, and non-self, which represents hidden dangers and invaders. Without a reliable way to decode exactly what's happening inside the cellular environment, our immune system would either completely destroy our own healthy tissue, or it would just totally ignore a lethal viral invasion. Neither of those is great. So, to solve this blind spot, biology evolved a brilliant solution. T cells can only see an antigen, basically a tiny fragment of a protein, if it is physically held out and presented to them. This process is called antigen presentation. It is the absolute core concept of our adaptive immune system. Think of it kind of like a molecular ID check. The T cell needs the evidence handed directly to it on a highly specific molecular platter. Section 1. The cellular billboard. To actually present these antigens, our cells use a tightly linked cluster of genes that code for a set of specialized cell surface proteins. These proteins make up the major histocompatibility complex, or MHC for short. In humans, you'll also hear this called the HLA, or human leukocyte antigen complex. Think of it as a literal billboard on the outside of the cell, advertising exactly what is going on the inside. Now, what's really wild about this is the sheer scale of the operation. We're focusing on the body's interior alarm, MHC class 1. This billboard is found on nearly every single nucleated cell in your body, plus your platelets. The only real exception here is mature red blood cells. That means almost every single cell you have is equipped with this alarm. Its whole job is to display endogenous antigens, meaning proteins generated from inside the cell. This includes normal cellular proteins, viral proteins if the cell gets infected, or even mutated proteins if the cell has turned cancerous. And the highly specific audience for this billboard? Cytotoxic T cells, also known as CD8-positive T cells. And this brilliantly illustrates the ultimate sacrificial nature of your cellular defenses. The fundamental message broadcast by an MHC class 1 molecule is simple, but it's pretty dark. It says, look at what I am making inside. If this looks foreign, kill me to save the rest of the body. It's an immediate lethal self-destruct protocol designed to halt an infection or a tumor right in its tracks. Section 2. The 5-step security pathway. So how does a piece of a virus hiding deep inside actually end up on a billboard on the outside? Well, it doesn't just happen by magic. It requires a highly coordinated cellular heist consisting of 5 distinct steps: degradation, transport, loading, presentation, and finally, scanning. By tracking this entire process, we can see exactly how a cell captures a peptide fragment, moves it all the way to the surface, and interacts with a passing T cell. All right, step 1 is degradation. Inside your cells, whether they're perfectly healthy or actively infected, cytoplasmic proteins are continuously being recycled. This is handled by a cellular recycling machine called the proteasome. Acting like a relentless molecular wood chipper, it can literally shred dozens of proteins a second, breaking them down into these short little fragments called peptides. Once the proteasome has chopped up these proteins, we move right to step 2. Transport. These freshly cut peptide fragments can't just float around aimlessly. They need to get to the manufacturing floor. So they're actively pumped across a membrane and into the endoplasmic reticulum, or ER. This highly secure transport is handled by a specialized transporter protein called TAP. Now that we're inside the ER, we reach step 3, loading. This is the crucial manufacturing moment. Newly synthesized MHC class 1 molecules are just waiting right here in the ER, and they have this specialized structure called a peptide binding groove. When a suitable peptide fragment arrives via the TAP transporter, the MHC class 1 molecule physically grabs it and binds it tightly within this groove. Bam! The antigen is now secured to the billboard.
With the billboard fully loaded, we enter step 4, presentation. The cell now has to get this heavy machinery to the surface. So, the newly formed MHC class 1 peptide complex is carefully packaged into a transport vesicle. It gets routed out through the Golgi apparatus, which kind of acts like a cellular post office, and is finally pushed all the way up to the outer plasma membrane. The billboard is now officially erected on the outside of the cell, proudly displaying whatever it found inside.
Section 3. The lethal handshake.
Okay, so the billboard is up, but now someone actually has to read it. Step 5 is scanning, and the tension here is absolutely immense. Passing CD8-positive cytotoxic T cells are constantly patrolling, basically checking these MHC class 1 billboards. They use their T cell receptors to scan the peptide being presented. So the crucial point is this. If the billboard is displaying normal cellular proteins, the T cell recognizes it as self and peacefully moves on. But if the T cell scans the billboard and recognizes a foreign viral fragment or a mutated cancer protein, an immediate strike protocol is initiated. This binary life-or-death decision is happening millions of times inside you right this second.
If that foreign fragment is detected, the CD8-positive T cell locks on and initiates the apoptosis protocol. The T cell unleashes a chemical payload, releasing molecules called perforins and granzymes directly into the infected cell. This toxic barrage forces the presenting cell to undergo apoptosis, which is a highly controlled, programmed cell death. The infected cell is completely destroyed, taking the hidden viral invaders or mutated proteins down with it, totally sparing the rest of the host's body. It's just a beautifully elegant, perfectly ruthless biological security system. A continuous, non-stop cycle of degradation, loading, and lethal scanning. But it leaves us with a lingering, pretty provocative mystery. If virtually every cell in your body is constantly forced to broadcast its inner contents on these MHC class 1 billboards, and if displaying a mutated protein guarantees immediate destruction by a passing T cell, then how do mutated cancer cells ever manage to avoid this alarm and grow into tumors? What biological loopholes are they exploiting to just hide in plain sight? Think about that for a bit, and we'll see you in the next explainer.